Good evening. Sergey mentioned I will be giving a brief introduction of the patch clamp technique, just simplifying or illustrating these results that you can have with patch clamp using halophytes for salt tolerant. So let's start. So the title is Use of Patch Clamp Technique in Plant Stress Physiology. So mainly, what is patch clamp? The patch clamp is an electrophysiological technique in which we are able to clamp a voltage of an insulated cell membrane or a piece of membrane. We will see this later. But by clamping the voltage, we are able to observe currents between this membrane and measure them. It has a high definition. You can measure even a current for one single channel, measured in picoamperes. That is a very small quantity of current. So, well, the patch clamper, the developer Sackman and Neer make it in 1981 with this work, improved patch clamp technique for high resolution current recording from cells and cell-free membrane patches. The importance of this technique is that it allows us to recording the movement of single molecules of ions in real time. So for developing this technique, they win Nobel Prize in 1991. Okay, so first, what is an ion channel? An ion channel is a transmembrane pore forming protein that help establish or control the voltage gradient across the plasma membrane, which we know as a membrane potential. It's allowed to flux ions thanks to the electrochemical gradient. So here we have an ion channel. The membrane is in pink and allowing ions flowing through this membrane. Okay, this is an open state of the channel, and this is a closed state. In a closed state, of course, no ion can pass through these proteins. So just to remember that it's important to have in mind that they could have basically two states, an open one and a closed one. So there is many types of ion channels. There are ion channels that are voltage dependent, that they open when they send some voltage in the membrane. There are other channels that are mechanosensitive. They can open with a stress or sensing some pressure. Now they have discovered some channels that open sensing temperature. And so like this, they send a stress, they open. There are other channels that sense a ligand. When a ligand coupled to these channels, they open. There are others that when they are phosphorylated, they go to open state. Well, ion channels has been found in all organisms, and in all tissues. They are involved in much of the biological process, such as membrane potentials, maintenance, regulation of cell volume, epithelial transport, translation of chemical signaling, cell cycle, and many other processes. In animals, they have central roles in complex processes such as thought, movement. Also in the nervous system, mainly all the signals are given through these channels, or thanks to these channels. It generates in the coordinated opening of these channels in the nervous system can generate action potentials, postsynaptic potential, or peacemaker potential in the case of the heart. So they have even found in virus, forming in virus, they are called viroporins. And these viroporins are very important because they lead the entry of virus to the guest, to the cells, and they can help to the replication of virus proteins and the membrane permeability. Well, an ion channel is not silly. Why? Because they can have, well, first, This is a structure of an ion channel. It's a crystallized structure. It was first discovered by Roderick MacKinnon. Again, he won the Nobel Prize because of this. The structure is very complex. They have charged residuals in the pore that can coordinate the selectivity of the transport of a channel. What is selectivity? Well, a transport not just conduct ion or any ion. They are very selective to conduct maybe one kind of channel just conduct potassium, or maybe is not selected that can conduct sodium, potassium, or other ions. In this case, in this image, it is illustrated that potassium can pass because it can unlock these 4 keys, if we can call like this. But sodium, even that sodium is smaller than potassium, cannot pass because the links that sodium has with water molecule is just 2. So the channel don't open or don't let pass these sodium channels. But it's also important that there are channels that are not selective, equally important, that just allow the flow, the flux of ions. Maybe if they can be sodium, potassium, chloride, any. Okay. So also the ion channels can interact with several signaling proteins. Here we have a calcium ion channel that is interacting with proteins that are in this inner side of the membrane or in the inner side of the cell. But also there is very important that they have subunits. We call a beta subunit. Beta subunit can also modulate the behavior of channel or how they could transport a different rate of ion that when it's not the beta subunit. But in the same time, a channel can modulate the signaling, making very, how to say, very ideal ionic conditions for the signalization. So in this case, let's put an example: the signalization of calcium. The signalization of calcium needs to have a very negative potential. So maybe some anion channels are opening, letting some negative charges to hyperpolarize or making more negative the inner part of the cell, or maybe some other channels that conduct potassium can open and taking out some positive charge and make it the same more negative, and this way calcium signalization can be done. And in the same time, calcium signalization can modulate the channel, so it's like a cycle. And this is the difficult part when you want to put all together. Okay, so this is the Apache clan equipment. We have an amplifier, which we use to stimulate the cells, and we have a digidata recorder or digidata card that receive these signals and traduce it well, this signal are digitalized and put it and so you can see it in the computer, and then you can measure. We have an anti-vibration system because that is very important, because imagine you are taking just one cell with an electrode. Any move can be breaking the seal you have. We will see later how I make the seal. You also have the chamber for experiments where you put the cells, and the manipulation system that helps you to approach the pipette to the cell. So basically that is Apache equipment. So what we basically do, we fabricate an electrode, and then with help of the manipulators, put it in contact with the cell. And then we apply a small suction inside the electrode, so we can have a seal. Here is illustrated how is a seal. So the electrode is in contact with the membrane, so we can form a seal of 1 to 10 gigaohms. When a seal like that, maybe the distance between the membrana and the electrode, it could be measured in dozens of angstroms, really small. So in this patch configuration, we call it cell attach. Maybe it's the more basic patch configuration because it's the first one you have. And from this configuration, we can pass to other configuration that will allow us to record other channels that are in the membrane. Because in this configuration, you just can measure the channels that are in this zone. That is between the electrode and this part of the membrane. So the other configuration is when you pull or you apply more suction, this part of the membrane breaks, and this we call that you have access to the whole membrane, so you can measure the current passing through the channels that are in the whole membrane, right? So then you can have an other configuration called this is outside. This is inside out. This has the inner face of the channel. You have it out in contact with the bath solution. This is useful when you want to prove that a drug is acting directly to the inner face of the channel. And this is the other configuration outside. This is also very important because with this configuration you can measure single channels, and this is important because you can find the conductivity of just one channel. And when you have this data of the conductivity of just one channel, what we call single channel current, you can count the channels of the whole membrane. Basically is what we don't counting channels, how much they conduct. This is a picture, a high definition picture of how patch electrode seems. And this is a transverse segment of the pipette. In the tip could be even 700 nanometers in diameter, so it's really small. This is the picture of a cell. This is human skeletal muscle cell that is about to be patched. So the pipette or the electrode is approaching to the cell, so we can make the cell attached configuration. In this picture we show a microelectrode in the cell attached configuration. Now it's attached, the seal is formed, and is all we need to start making the records. Let's say. So basically, I don't want to talk much about this, but basically we can see this system as a circuit. So we have the pipette, we have 3 resistors, 2 are in series. So the this resistance could represent the pipette or the microelectrode. In series we have the resistance that could be the resistance or access resistance that could be the pore that you make when you break this part of the membrane. You have also the leak resistance, that we want this resistance is as bigger as possible, because when this resistance is low, it won't allow us to record channels. So we need big seals to make this resistance very low. So we can see all the cell or the membrane as a capacitor, and the channels we can saw it as a resistance also. And So what we are recording is the current that passed through this resistance. How? If we have many channels, well, there is several equations that they use to approach to this, but in a basic form, any channel, when it's the same type of channel, has a conductivity, and this is a conductivity that has the resistance. So if we have two channels of the same type, we can say that the conductivity of these two channels will be 2 multiplied by the conductivity of a given conductivity of the channel. This way, we can count how many channels we are measuring in open state in any experiment. Let's say that. So the summation of all the conductance tells you how many channels you have working in that moment. Okay. So any question in here? Because now, really, I think then you are understanding. I will ask later at the end. Huh? Okay, we use an halophyte species to see how these channels in the vacuole are behaving. This is just for studying the salt tolerant, let's say this way, the molecular basis of the ion channels basics of the salt tolerant. So we use Chenopodium quinoa, that is a very well-known halophyte. We use 3 varieties of this: a sensitive one that we will call Q52, an intermediate that is Q20, or tolerant that is 16. Well, all these plants were watered in daily basis during 3 weeks in control using 50 millimolar of sodium. This is because they naturally need sodium, and in salt condition that they were watered with 400 millimolar sodium. Well, to get the protoplasm, I take leaves. I take upper leaves, that this is the young leaves, and older leaves, that is the leaves that are in the bottom. I cut it, I peel it, and then I use an enzymatic insulation. So then with centrifugation, I could obtain, this is just a brief explanation of the enzyme protocol. And when I obtain the protoplasm, I put it in the chamber so I can start working with that and using the micromanipulator to take out the protoplasm membrane mechanically. So I approach the microelectrode and then pull a little bit so I can break this membrane, and then I can have the released vacuole. And I was working in the released vacuole. This is the rest of the protoplasm membrane, and then I try to make the ceiling here in the vacuole so I can record the currents of the FB channel and SB channel that I will explain now. So basically, what an halophyte wants? A halophyte wants that, well, this is I think you know this model is a Sergei model that proposed for the ionic and osmotic components in solid stress, but I use add here. What an halophyte wants is to maintain the excess of sodium in the vacuole. Of course, there has to be some small amount of transport to the cytoplasm, but not much, because they always have to have a ratio for the normal physiological process take place. But in excess, sodium will be very dangerous in this part, in the cytoplasm, because maybe it will start kill organelles, or maybe signalization process won't be done it right. So the important thing is that they have to maintain sodium in the vacuole. Well, that is the hypothesis. So what I did, then I took a vacuole, as you can see it. I use certain conditions, and with the amplifier, with the Kidman I show you, I send some protocol of voltage. We can see it here. Like I start in minus 180, and then again I were placing another voltage that it was minus 160, and in red I show the voltage that I am applying for stimulate the current to plus 120. We can see in here when we apply the voltage, we can see in red that is the current that is stimulated because this voltage. So I apply the voltage and I read the current through the membranes that I have here. So this current, if I apply the voltage in here, this current is given in here. And I am reading it with other electrode that I have in here. We call it the reference electrode. So anytime I apply a voltage, I have different current, right? So this is an FB current stimulation protocol. With this protocol, we can see the FB current that is fast bipolar ion current. We call it fast because, well, you can see it better in here. Because this, to get the steady state of the activation, is really fast. Well, sorry, it's fast. In difference with this, that is the slow. So channels are open in a very slow way, fashion. And in here, when you apply the voltage, they are already open. And what in this protocol tell us that the channel slow backward current SB channels are voltage dependent, because if we apply any voltage like in here that I apply 100 millivolts, 80 millivolts, the current is zero. You can see it in here. But as soon the stimulation protocol is going, I am applying negative voltage, the current is stimulating. I am generating current. That's why, because these channels start to open maybe at minus 40 millivolts. So once I start applying minus 40 millivolts, the channels start open. So these kind of channels are very different, and both of them we have it in the vacuole, but not at the same time, because if we add some calcium or magnesium, maybe an amount of 1 millimolar, we can inhibit this channel and start having these kind of channels. And when we watch calcium or magnesium, we start having this other channel. So these currents, I just measure it and then I analyze it, and I can have this. This is, we can call it that I comparing the level of current with the voltage at which this current was stimulated. So then after I process this data, I could have the conductance of the channel in any situation. I need to. Yeah. So what do we have here? We have the FB current in quinoa 20, that is an intermediate state between the salt sensitive and salt tolerant. So we use young leaves in a control condition, and we graph the data. Okay, well, sorry. In here I have two states of the. In here I have two states of the channel. This is the initial state that we can use it as a more physiological state because it's at time zero. And after a time, this channel wash all the components that are in the cell wash. So maybe for biophysical, this is a more, let's say, pure state, so they maybe want to use more this kind of analysis, but for this seminar I will use the initial because it's more physiological, and we will focus in this part because in this part is the more physiological voltage. In physiology, in natural state, we never will have voltage like 100 or minus 100. We always will have maybe from minus 10 to 10. So we will focus in these results. So what I did here, I am comparing a young leaf in control condition with a young in salt condition. So basically, it is the same conductance, but then when I was comparing all control or on salt, the conductivity is lower. What this is telling us that maybe in all leaves the sodium is concentrating, not that, and the vacuole doesn't allow to go out the sodium. So this is not new. We know that in a strategy for plants is that concentrate sodium in all leaves, but at least this is maybe one of the first evidence that this is happening. And how? Using these kind of channels. So this is an analysis of the other type of channel, SB. It's very different because I mentioned it's voltage dependent. So they start to open maybe a minus 70, as you see here. Here I representing the current, and it happened the same. For young control, I have a bit current. Maybe I can express this in number of channels. Oh, let me see. Okay. Maybe in here I present the graphic that is the current between picofarads and between the conductance of the channel. That give us a number of channel open in a certain voltage. Like here, at 0 millivolts, we have for these two conditions, the young salt and young control, we have near 20 channels open in open state. But for all in salt condition and all in control condition, we have less channel open at this millivolts, at this voltage. So what does this tell us? That it tells us the same. In this moment, the all plants, the vacuoles in the normal plant doesn't want to take sodium out. So in this way, they are storing sodium and just maybe like making this sacrifice to store the sodium and protecting sodium doesn't go to young leaves or well something like that. Well, that's it, and a strategy that has been proposed for many times, but maybe this is molecular evidence of how this is happening. Oh. And then I compared three different varieties of quinoa. One was the sensitive, the intermediate, and the salt tolerant. So what I have in here, that the behavior of the channels, of the opening of the channels, are really different. Having than the salt tolerant quinoa has channels that conduct less ions, as we can see in here. This is the tolerant condition. In here we can see the intermediate, and then the sensitive. And the characteristics is really logical because we analyze that the sensitive variety has at 0 voltage has 27 open channels. For the intermediate variety has 20 open channels in here. The tolerant variety has just 9 open channels. The same happened for the FV current, where we have the sensitive variety has 1.5 higher conductivity than the tolerant in control condition, and is 2.9 higher than the tolerant in salt condition. So what does it mean? Also, for control in the tolerant variety, the conductance of these channels is lower than the sensitive one, and it's even lower when you treat it with salt. This is just the relation between these two curves, so I have to do this relation to get this data. But so, well, and by this time we think that maybe we can try some osmolyte or some components that could regulate the channels. So we know that betaine is very important in the halophytes, in the salt stress tolerance. And then there is a publication, a recent publication. I don't even know the month. I just see the DOI. But in Sergei Chabala's lab, they show that exogenous application of physiologically relevant concentration of glycine betaine substantially mitigate oxidative stress damage to photosynthesis too, in the dose-dependent manner. So I try betaine in these channels I just tell you. And now let's see the result. For sensitive, we already know this, the conductance is higher than the others, but when I use the betaine in the same condition, in control condition, in a sensitive variety, this takes, the betaine take the channel behavior or the conductance of the channel to a very similar states where that the tolerant variety, as we can see in here, is the same curve or the same data, but in here is with the betaine, and it's very similar to the tolerant variety. So what we have here, that maybe betaine could be a component that could be really important for the behavior of the channels in this tolerant, in salt tolerant. So even when we add betaine in the control condition for the tolerant variety, it takes even the conductance of these channels even lower, as we can see in here. So let's put it in a quantified way. Is the conductance of the sensitive with betaine is 0.85 well lower than the tolerant variety in control conditions, and then the sensitive variety channels are 1.4 higher than the tolerant conditions. In the other way, it was almost three times higher. So betaine, it takes to a lower conductance. So more similar. Now with betaine, so we can conclude that with betaine, the sensitive channels or the channels of the sensitive variety behaves like a tolerant variety. So in here we are comparing in salt condition the tolerant variety, and what we saw when we compare the salt variety conductance with the one that has betaine, well, nothing happens. It's maybe because betaine just take reach a limit, so it doesn't take it more or more lower, and maybe, I don't know, other components are are implying in that. I'm not saying betaine is just all. Maybe some other behavior we haven't seen, or other components. I are also trying other osmolytes that are important in halophytes. Just I don't showing the results, but I having good results for that. And well, maybe that's all until now. So what are the conclusions of this work? Until now, I cannot make a main conclusion because we have to compare this data with data obtained with MIFE, with data obtained with intracellular measurement of calcium. But just small conclusions that we can have is that FB and SB channels have an active role in the adaptation of salinity by altering properties in its own leaves. Then there is a significant difference in the conductivity of FB and SB channels between salt-sensitive and salt-tolerant varieties of halophyte plants, and then that betaine can modulate vacuolar ion channels behavior of a sensitive variety to stimulate the ion channels behavior of a tolerant. To simulate. And then also another conclusion is that maybe it's the first time we are showing the betaine effect on ion channel, because there is not much people doing this technique in plants. Maybe 15 labs in the world are doing this, so there is a big area that you can use it for making some projects. Next, I will explain maybe some other application this technique can have. So just last year they found, or they make this model that vacuoles, for not going just further, that vacuoles are implied in cell death in two different ways. Well, in these publications they never talk of an ion channel. They don't even want to talk about ion channel, because they just don't know nothing of ion channel. But I know that, imagine how vacuole is modulating the volume in this process. I mean, this could have a huge movement of ions in this. So of course ion channels has to be involved in that, but we haven't proved that. So there is an unexploited area that you can also use this technique for maybe make some projects of that involving in cell death. This is another that they show. They took some picture with the vacuole fusion to the membrane, forming some pores. Well, of course, not selective pores, and they propose these pores are for getting defense protein, but for getting out defense protein against bacteria, or even hydrolytic excision for against virus. But the thing is, for making all this process, there has to be some regulation of ion channels, and nobody is mentioned that. So it's let's say like an unexploited area for also making some research of that. Another aspect of this is that in plant signalization there are many pathways that hasn't been really explained. Well, in here that can be a secondary protein, but modulating to this channel. But really, how these channels are helping to help the ideal ionic conditions for all these pathways happen. Not much information is known in that. So, but you can have also these proteins that they are now really important, because, well, not important. Well, I just find that these proteins are important in the biosynthesis of alkaloids. When they were trying to accumulate alkaloids in plants, and I find these proteins has interacted with some channels in cardiac myocytes and in neural cells. So why, if also in plants these proteins are modulated with channels? We don't know, and this is also an exploited area. Also, as cancers, I mean, in animals, this technique has been used a lot to find to see how channels are involved in tumorigenesis, or maybe in some regulation of arrhythmias. I mean, all drugs, the therapeutic drugs are having the target in channels. Well, maybe 80% of the drugs, but they just don't know exactly the molecular basis for that, because they started in an empiric way to prove these therapeutic drugs in animals, but then they were finding that he has his primary target in an ion channel, and now they are making these new drugs based since how they interact with the channel to develop this new pharmaceutical target. So.